Welcome to Bug Talk, a podcast hosted by the Entomology Department at Michigan State University. Today, we are talking to Dr. Eric Benbow, who is a professor of aquatic and decomposition ecology at Michigan State University. His work highlights the broad applications of entomology with a primary focus on how insects and microbes interact with each other across a variety of settings, specifically in the ways that disease spread, decomposition, and even the ways that insects can be used for food and livestock feed in Africa. So, could you tell us a little bit about your background and what led you to doing the work that you're doing today?
So my background, I'm from Dayton, Ohio, and did my bachelor's degree at the University of Dayton in Ohio, and then switched into graduate school there and finished my PhD. And while I was doing some field research in Hawaii, Dr. Rich Merritt, who used to be the aquatic entomologist faculty member here at MSU in entomology, he was visiting Maui. And long story short, we ended up meeting, and he had a postdoc position open at MSU. And so I needed a postdoc job, and that's how I ended up at MSU in aquatic entomology, aquatic ecology. But also Rich was deeply involved with forensic entomology. And forensic entomology is how you use arthropods in a court of law, right? And often that's associated with determining the time of death in a murder investigation, as an example. But what I realized, helping him out on some forensic cases here in Michigan, was that there was very little information understood about the decomposition of dead organisms, like deer or pigs or rhinoceros, for instance, in Africa. And so I got more interested in what the ecological processes were associated with that, and then how you can use that information to better understand, death investigation and the applications of forensics. So it's kind of how I got here. And in the meantime, I was also studying aquatic ecosystems and aquatic entomology. And Rich also had a new project in Africa that we were trying to determine whether or not aquatic insects were responsible for the transmission of a mycobacterial pathogen. And that's where I actually got my first interest and exposure into understanding microbial ecology of aquatic ecosystems. And so those two lines of research, when I came to MSU as a postdoc, really have moved my career in this very dynamic and interdisciplinary areas of research, where understanding how insects and bacteria, or insects and microbes interact in aquatic ecosystems for disease transmission, or in decomposition scenarios for carrion, or how you might use that information in forensics. And then insects for food and feed seems like an interesting tack on for a lot of people. But the insect that we're focusing on is the black soldier fly. And that insect is also known to colonize carrion and decomposing vertebrate carcasses. And so that particular species will also be very efficient in decomposing vegetable organic matter, or brewer's waste, or waste from restaurants, the food waste there. And so it's kind of associated, but the insect microbe part certainly goes both ways. And so that's how I've gotten to this kind of strange situation of a career.
That's awesome. Could you explain what insects have to do with decomposition and forensics? Like, I know that you said it can help you figure out the time of death and everything, but like outside of that, how do they connect?
Well, I mean, if there weren't evolved insects to complete their life cycle and feed on carrion tissue, right? Naturally, carcasses would just simply build up, right? Mm-hmm. Bacteria and other microbes, they contribute to that decomposition process, but it takes much, much longer. And we know this through experiments, if you don't have necrophagous insects that colonize the remains. And so is an important ecosystem function. A lot of the insects that have evolved to detect and then develop on carcasses um, inherently play a pretty important role in ecosystem processing, basically generating recycling energy and nutrients from the once living organisms. The microbes are pretty interesting with carrion. We think that the microbes produce volatile organic carbons, and so those VOCs, um, we believe, and there's some evidence to demonstrate that they attract necrophilous insects, and that attraction then brings in certain species that then will oviposit and lay their eggs on the carcass. And then we also think that those blowflies will bring in their own bacteria and microbes and basically seed it. And some of the hypotheses are associated with if you seed your resource with your own bacteria, it's going to provide an advantage to your larvae, your immatures, as they develop and grow and consume those bacteria, and then the bacteria proliferate. So on a carcass, this ephemeral resource patch that is highly sought after for many scavengers, both vertebrate and invertebrate, the microbe insect interaction seems to be very important in terms of recycling that resource. And then the way you can use that in forensics is a couple ways. One, because the insect life cycle is predictable based on temperatures, you can determine how long they've been colonized by the insects, and that's often pretty close to the time of death. But two, as the microbes change on the carcass, we found that we could use microbial communities as kind of a microbial clock to also understand how long the once living organisms has been dead. And then another way is microbes and insects will leave behind evidence, meaning microbes change the soil, microbial communities, and chemistry of the soil right underneath the carrion or the body. A lot of the blowflies will migrate from a carcass to complete their life cycle. So they go to find a nice safe place to pupate, go through puparation. And so if you move a carcass or if you move a body, there may be evidence to suggest that the body was actually at a different location, and then you move it somewhere else, and that provides information to the investigators on maybe we should ask different questions in this investigation of our potential suspect. So there's a variety of ways that the basic understanding of carrion ecology and insect microbe interactions can inform forensics, and I just kind of mentioned a couple of the big ones.
Thank you. Can you explain what disease ecology is in the context of your work?
Yeah, disease ecology it's part of a One Health concept in many ways, really trying to understand what are the environmental roles of organisms and abiotic factors that contribute to the spread and emergence of new pathogens that cause disease in humans and other animals. So for instance, some of the hypotheses that we've explored are when you have deforestation, it affects water quality conditions in certain places of Africa. We think those water quality conditions then change the community ecology dynamics of macroinvertebrates, fish, and the microbes. And that shift in that network of organisms, we think, and others have shown, will increase the likelihood of transmission of a pathogen to humans or wildlife. Lyme disease is another really good example of trying to understand how the different hosts of different types of ticks will lead to greater tick abundance, and then the likelihood that someone walking through a prairie in long grass will basically be colonized by the tick, parasitized by the tick, and then the pathogen will be transmitted to them, and then they'll present disease. And so there's some really fascinating work from New England that shows that masting of acorns affects the differential abundances of mice and deer, and those changes then affect the overall risk of having more ticks that will potentially parasitize humans, and therefore you get disease emergence. So there's a lot of different examples. Vibrio cholerae Furnunculus from the south, trying to understand the aquatic ecology of Vibrio and how that can lead to disease in humans as well. So it's the ecological dynamics and the evolutionary history of those organisms associated with that disease system that defines the disease ecology of specific diseases.
Okay. I was on your website and I couldn't help but notice the a lot of information about the Novel Weapons Project. Could you give some more detail and like explain what the Novel Weapons Project is?
Yeah, so the novel weapons hypothesis, we kind of borrowed it from plant ecology literature, and that's basically invasive plant ecology literature, where one of the explanations or the hypothesis that invasive plants establish so well in a new environment is that they harbor novel weapons, or they harbor the ability to make certain chemicals that allows it to outcompete other plants that they would normally compete with and keep them at bay, right? To keep them at low population numbers, or maybe even not allow the invasive or the new species to take hold. And so there's a lot of interesting literature. It's not universal, but there's some good examples where there are invasive plants that have come in and they drop certain chemicals from their leaves into the soil around them, and that prevents other plants from establishing. A few years ago, when we were thinking about our disease system, related disease that most often is found in children in Africa. The epidemiology is a little bit different in Australia. Older people tend to get it. They have different activities, but it eats away at the skin, and it leads to huge disfiguration and often amputations and severe morbidity. And while we were studying that, one of the ideas was, well, the pathogens, Mycobacterium ulcerans, and it shares relationships, phylogenetic relationships with other pathogens like tuberculosis and leprosy. And part of the epidemiology and the way that it affects humans is it produces mycolactone, and this is a toxin. We call it a toxin because the clinicians and microbiologists identified it as the molecule that causes the disfiguration, the eating of the skin. It's cytotoxic, right? So it kills cells. It's immunosuppressive, so you don't really feel much of it for a while until secondary infection by other bacteria set in, and then you start to present symptoms. We started thinking about what's the actual role of mycolactone in the environment, because we were able to detect that pathogen in aquatic invertebrates, aquatic insects, and fish, and biofilms of plants. And so you find the pathogen in the environment all the time, both in Africa, in Australia. We've actually discovered it here in North America. And so the idea was, well, what if it actually is a weapon, a novel weapon that allows it to persist and disperse in new aquatic habitats? So let's say it gets on to a bellostomatid, the bacteria does, and that bellostomatid flies 200 meters, or hops, multiple dispersal events to get 200 meters, maybe a mile away, into a new aquatic habitat. Now it brings the bacteria with it. We hypothesize that bacteria now upregulates that toxin to allow it to take hold, much like the plants, right? It's getting into a new environment. It has competitors. Does it upregulate this new toxin in the environment? Now, if you have a huge abundance of that pathogen and they're all upregulating their genes for this toxin, and then a human goes in to do laundry or to go fishing or to do some other agricultural duty, and they have a cut, like a deep wound, or maybe they get cut by something in the water, does that get into their skin? And then the toxin, right? The molecule now becomes a toxin because it starts to destroy cells in the human epidermis. And so that's where the novel weapons hypothesis within our disease ecology work kind of originated, and we were able to get a good grant from NSF to study the ecological dynamics of aquatic invertebrate communities, plant communities, fish communities, other microbial communities, and run some experiments to determine if that mycolactone actually does influence other microbial communities, and that does that influence how it persists in macroinvertebrate communities as well. Then we coupled that with another hypothesis called the coincidence virulence hypothesis, which is more associated with disease networks and disease systems. And what that is there's some parasites and pathogens that haven't evolved to become pathogens. They haven't evolved with their host, right? But they just have the molecular machinery or the life history that allows them to cause disease in a vertebrate, human or otherwise. And so it's just a coincidence of their evolution that they can also then create disease in humans. So we coupled those two hypotheses to understand maybe it's a novel molecule, novel weapon. And so they use that as they would in any ecological circumstance, but now the bacteria finds itself in skin. And so it's coincidentally causing pathogenesis in the host, if that makes sense. And so that's kind of the combination. It's a deeper ecological invasive species question that we've kind of fused with some more modern day epidemiology type of terminology.
Okay, and just to clarify, what is pathogenesis?
Pathogenesis is the ability or activity of some, usually a bacteria or virus, to cause disease.
Okay, okay.
And you
And
present
then,
symptoms.
and then what's the disease that like eats away at the flesh? What was that called?
Buruli ulcer.
Okay, okay.
Buruli, ulcer, ulcer.
Okay, okay. And then from my understanding, all this research is being done in, correct me if I'm saying this wrong, French Guiana?
Yeah, currently we finished the project there. Currently, the earlier work was in Ghana, West Africa, then moved to Benin, West Africa, and then the newest grant was in French Guiana in South America. So that's where it's been mostly recently.
Awesome. So what brought you guys to French Guiana? Was it just like that's where the disease is mostly found, or how did you guys end up overseas?
It's actually much more abundant in West Africa.
Okay.
West Africa is much more difficult to work in the field. And part of that is the infrastructure that's available. Part of that, and it's a real concern when you work in waterways, major predators. We're in crocodile habitat a lot. Some of the most venomous snakes in the world are in West Africa. The logistics of getting around and doing some of that field work is very challenging, very expensive. And when I was presenting some work at the World Health Organization in Geneva, there was another Buruli ulcer researcher there, world-renowned disease modeler, mathematical modeler. And he saw my presentation, I saw his, and we had a cup of coffee later to discuss some ideas that we had. And coincidentally, he is French and has done a lot of work in French Guiana. I was very aware of his work, disease ecology, mathematical modeling, very, very, very cool stuff. And we decided, maybe we could collaborate. And so we shared some data with him. He modeled it. We got some papers. And then I said, can I come over and visit? And can we hash out a new grant idea in French Guiana? And so while French Guiana doesn't have a huge amount of disease right now, we know from his work that the pathogens there has been a lot of disease in the past. It has gone down considerably, and that's kind of how it's been everywhere but Australia over the last 10 years. But he said, okay, well, why don't we go visit French Guiana as well? So I said, okay. And so he invited me to give a talk at this LabX conference that is always hosted there. And I immediately realized how much easier and practical it could be to work in French Guiana. And I say that because if you've never been there, it's basically a French territory. It's a French district, arrondissement, I think. Um, and so the European Space Station is there. So if you can think about the infrastructure necessary to maintain a not space station, a launch pad, space facility, the infrastructure is really important. And there's a military there. Um, there are caiman, right? They're not as big as some of the crocodiles we saw in Africa, but there's venomous snakes, but there's only one that can really do some major damage. It can kill you, but it's not often killed. In Africa, a cobra, a mamba, you're dead in like 20 minutes. So it became really clear for a couple of reasons. Infrastructure, fewer predators, makes it a little more safe, but also in French Guiana, huge amount of the landscape has not been disturbed. Or if it has, we know when, like illegal mining got into certain areas. They have a lot of the European scientific community has a lot of information on that. And so it seemed like a really opportune place to study the ecology of Mycobacterium mulcirans, study the ecology and evolution of other mycobacteria in French Guiana, where we could get to places much more easier than we could sometimes in Africa, and with the infrastructure that allows for a lot of this, and then the more pristine habitats that were really well studied and really well known.
Okay. And then with you working like overseas in areas where like there's a lot of dangerous wildlife, do you have any like horror stories about like anything that's gone completely wrong?
Well, all of us never had a medical emergency. We'll put it that way.
Okay.
There were times early on in the 2000s that I think you're standing in some waist deep water and you're sampling aquatic invertebrates, right? And something drops from the tree, 5 feet away, and your colleague says, I think that was a mamba. And you're there with hosts, right? And African hosts that they know the species there, and they're like, yeah. And so you just stand there and you're like, what do we do now? You wait it out, and then you slowly kind of move your net around way far from you, and kind of hopefully it moves out, and then you slowly get out of the water. But to be honest, I think the scariest part of that, and I think anyone who's done field work in Africa, it's driving, and driving at night is, one of those logistical things that you don't want to be on the road at night. And so let's say you're 3 hours into the field from your lodging. You only have about 3 or 4 hours of light before you have to start driving back if you want to be back before dark. And so sometimes that just wasn't available. Car breaks down. Now you have to stay somewhere, way in the bush. And to get to this next small village, you have to be on the road for an hour. And some people ask, well, why is that? Many people there, they don't have the resources. And let's say that they don't have front headlights that work. And they need to be able to get to their next destination before the next morning so that they could provide any kind of resources for their family. And so they're on the road without headlights. That's just one example, but it's real there. And amazing people. I mean, I have so many good friends from there, and I've learned so much about cultures and the situation in some of those areas. But yeah, there's the real challenges associated with that. And one other story, I had a colleague that was walking down a trail and must have gotten into some ants. Literally had ants in her pants.
Oh no.
It was
horrible.
both funny because she wasn't allergic and she didn't get really bitten much, but it was a very precarious moment for about 20 minutes. So we let her clean up, and we went back to a village and waited it out. But I'd never heard of someone literally getting ants in their pants. But she started dancing, and we didn't know why until she screamed a little bit. But she was fine and took it in good
Okay, good, good,
stride.
good. Do you know what kind of ants they were?
No, no, they weren't army ants because we've seen a lot of those, and I've stepped over lines of those before. You don't want to be army ants, and they must not have been fire ants because she would have likely had much more severe reaction. It
Right.
was just annoying more than anything. Um, so I stepped over that ant line.
Yeah. And then when you're out in the field, like day to day, specifically, like what do you guys do?
Oh, if you're interested, I can send you some videos.
Absolutely.
My postdoc actually made a video of our work in French Guiana, basically day-to-day activities, from camping in the middle of the rainforest without any infrastructure at all, having to make um, your tarp-like tents out of the resources that were there. And then you get up and you get in a boat and you take all of your material to your sampling location. And there's three teams. There was a microbe team from Mississippi State University, a fish team from Mississippi State University, and then a insect microinvertebrate team. And so we would do semi-quantitative, standardized sampling of aquatic invertebrates, pick them in the field, preserve them in the field, um, help out with other measurements at the location, like water depth and water quality measurements. We go help with some of the fish captures if that was appropriate. We also help with some of the microbe. While in the field, you'd make up your own structure. If it started raining, because it's a rainforest, it rains all the time, and we would sit and start picking some of the insects and under these makeshift little tents type things, um, and bring everything back. And we spent 4 weeks there one year, 5 days in the deep rainforest. That was one of the most profound experiences of my life.
Yeah, I can imagine.
And oh, you want to hear one more story?
Yes, I do.
From French Guiana. This was in Africa. This was something completely unexpected in French Guiana. One of our last trips. Um, so we didn't always sleep in the deep rainforest, but there was often these large, more permanent structures where you could put your hammocks. So there are about 9 of us. We put up all of our hammocks. We're used to this by now. That's how we lived all over French Guiana. And I was on the end because it's cooler, right? On the end, because when there's 9, people on the end get the coolest, people in the middle are the hottest. So, you try to get people to get cold. Well, in the middle of the night, I had something tugging on my foot in my hammock. And these are parachute type of hammocks that most of us were using. And I'm like, it's never happened before. Why would it be snagging? No, I went back to sleep. Woke up again, but something, it almost hurt my foot. I'm like, what would that be? And so I put my hand down, pitch black, right? New moon. And it's sticky. And so I put my headlamp on and it's blood all over my hand, all over my foot. And I'm like, what just happened? And I still didn't know. It was about midnight. Other 8 people are sleeping. I'm like, no, it couldn't have been anything. It couldn't have been a snake. I'm hanging up. We have these barrier things on our hammocks. I'm like, 3 feet up. If it was a jaguar, I'd be dead. I mean, it wouldn't just snag my foot. I started thinking about it. I mean, there's vampire bats all over the rainforest.
Oh no.
And they will attack cattle at night. So when you sleep in these parachute type of hammocks, the ones that you see all over campus, right?
Mhm.
When you sleep in them diagonally, and when you do that, you push out like it looks like a bulbous structure. Well, my foot looked like, I think, the hip of cattle. And it had started feeding the first time, and they produce these anesthetics, so you don't really feel it, right? And they also produce something that doesn't allow you to clot. So the first time I just didn't feel it. I just kind of woke up and it was nothing big. But the second time, I think I moved my foot on accident. And when it did, I felt the fang like slice.
Oh
And that's what
no.
really woke me up. Um, yeah, so I got up and I'm like, this won't stop bleeding. So we had some really high percentage ethanol. I'm like, if I got bitten by a bat, I need to clean this sucker out really fast and effectively. Next morning, I didn't tell anyone because I'm like, you know what? I'm on the edge. Um, if they're going to come back, they're going to come back to me anyway, and it's just going to wake everybody up. And we had a sample in the morning. So I sat there for about an hour, putting pressure on it. Finally, stopped bleeding. And the next morning, like, yeah, we heard you, Eric. You're up. We just thought you couldn't sleep. I'm like, yeah, it was different. So all the time, this is the best part of the story. All the time in Africa, like 15 years in Africa. I spent probably 2 years of my life in Africa doing field work there. Another 2.5 years in French Guiana. I'd never had to call my physician wife on the satellite phone. Never had to.
Mhm.
So luckily there was a helipad near our place where we're staying. So I climb up the next morning, top of the helipad. I call her. She lets it go to voicemail.
No.
20 years, right? I've never had called. She's like, okay, you can wait. So I call back. She's like, well, I probably should answer it. He's bothering me. She was seeing a patient at the time, but she's like, I got to step out. I'm like, well, I'm about to cry. And I said, I'm making this phone call. I'm 3 hours from the port, and then I'm another 3.5 hours to a hospital. And here's what happened. I told her, she goes, oh, we treat kids for rabies all the time. You'll be fine. I really got to go.
That's crazy.
Like, okay. So, so I actually spent we finally got back to Cayenne, the major area there. I was able to go to the hospital with my colleagues who spoke French, who could have been really difficult to get by there. And for the next 4 days, I took the European medical regime for rabies while everybody else went back out into the field. Um, so that was part of the remote aspects of this. I had 3 days before, if I did have the virus, um, before it was too late, and we were able to finish our sampling. We stayed at another place about an hour and a half down the river at another cavette, and then the following morning, so 2 mornings later, we made it back to the hospital, and then it was Sunday, so they weren't open, so I had to wait one more day.
That's so scary. That's horrible.
I did some research. I'm pretty sure I'm the first American to have reported uh, vampire bat biting, probably in the last 100 years. There's probably been others like me sleeping in one of those types of hammocks. Then so there's a rabies office at this hospital, right? Because there's lots of dogs with rabies and kids get bitten all the time. And I'm looking at this sign. It's all in French, but there's one with the circle with an X through it, and I'm like, that looks like my hammock. And next to it is another circle without the X through it, and it's a more traditional hammock that is cloth, really big, and it has a mosquito net over it. So we started talking because my colleagues there have people who study the vampire bats, their biology, their rabies frequency and stuff like that. They always use the traditional nets with the mosquito nets around it. Well, it looks like this to bats, it looks like this giant bulbous like block in the way they sense things.
Right, like a cow. Yeah.
Well, no, that's what I looked like. When you sleep in the traditional one, it's bulbous. It's just like this giant block.
Okay.
If you sleep in the parachute ones, you make projections with your feet and your shoulders and your head that look like cattle, and they're about the same size and off the ground as cattle as well. So I learned some things about camping in the rainforest. I will always use traditional nets with bed nets over them in the future.
Oh man, I'll keep that in mind if I'm ever in Africa, for sure.
Mhm.
Just out of curiosity, you mentioned like the European rabies treatment. What did that entail?
Um, It wasn't that bad actually. I had to get the immunoglobulin on the near the site where the bite had occurred. And then I think I had to take 2 shots every 3 days or so, something like that. And so luckily I got it all done before having to come back to the US because my wife she actually then started thinking maybe more serious. She started investigating here in the US about how I would have to come back and take the regimen. She knew the US medical treatment for it, and it's like weeks long, not just like 7 days or 9 days or whatever it was. Um, Turns out the US FDA had not approved the European treatment for rabies, although they both actually work just as effectively. Um, and so I was able to um, get actually the most effective treatment and the best treatment because it only lasted like 9 days total. And it worked out pretty well that way. And then I made sure that I kept cleaning it out so I wouldn't get any other infections from the bat. But yeah, rabies is 100% mortality. There's been like
Yeah,
one person who survived in the history of it. It's crazy.
you know. I mean, not saying you're lucky you got bit, but you're lucky you got bit outside of America.
True. Guess when I got back, like a week after I got back, my wife and I were hiking down a trail near our house, and there was a raccoon out during the middle of the day. Should not be out in the middle of the day. It's often a sign that they have rabies. You know what she
Mhm.
said to me? She goes, "You just got treated. You go first." I'm like, makes sense.
Yeah, she's offering up the sacrifice.
So anyway, that's my biggest story from French Guiana. But I can send you that link that my postdoc made of kind of a typical day. We also had a big motion picture documentary that was uh, made from Arte. It's a European branch, but it was actually shown in Japan, all over Europe, and I think there was a release here in North America at some point. I can't remember when, but I'll send you that link too. It's pretty cool.
Absolutely, absolutely.
Mhm.
And then while we're on the topic of Africa, um, in a Q&A article that was published back in February, you said that your research encompasses the community ecology of insect microbe interactions in several contexts, including insects as feed and food systems in Africa. Can you elaborate more on like the feed and food systems, how they're being used? Is it more for like livestock or for people?
Hmm. Um, Interested in both, right?
Mm,
In many parts of Africa and other parts of the world, insects have been historically consumed. A variety of ones, crickets, grasshoppers, and stuff like that. But they have to be harvested. And so they're
mm,
they're based on seasonal abundances of them. We've been most interested in black soldier fly, and that is primarily used for feed, for aquaculture, and for cattle and other livestock. Um, that is, the species Hermidia lucians um, will colonize carrion, but I really got more involved with a colleague from Texas A&M, Jeff Tumberland. And he's kind of a world expert on the black soldier fly and how it can be used to produce feed for livestock and aquaculture. Um, he had a very good focus on at large scale in North America and throughout the US. In Europe, has many collaborators in Asia as well, where they're trying to produce tons and tons each week. Um, I was able to recruit a postdoc through the African Alliance for African Partnership, AAP, a few years ago. And Dr. Deborah Ummelin came here to MSU. Unfortunately, got her studies going on the black soldier fly and using some local resources to understand growth and development of the insect. But COVID hit, and unfortunately, she had to go home early. Um, However, we kept continuing to work with Dr. Jen Peckall, who's an assistant professor here in the department. Got some money to promote developing black soldier fly at small scale, like farmer scales. And now Deborah has trained like 5,000 farmers in Uganda. Um, we had a symposium in May or in June basically bringing together 10 countries, farmers, testimonials. We sat around trying to develop ways that we could expand this training program, and that's where we're at. We're trying to ultimately impact over 1 million farmers in Africa, training them on how to use black soldier fly, not to generate hundreds of tons a week, but to generate enough to offset the feed costs for their pigs or for their cattle or for their chickens, or to produce bigger, better catfish or tilapia in their small scale aquaculture ponds. And so we're trying a different approach, not large scale industry scale, but more grassroots, um, where you get high densities of farmers using black soldier fly as feed. And so black soldier fly, what the really interesting thing is, it's really good at waste management. It recycles organic matter that's produced, like I said earlier, through breweries, brewery waste, distillery waste, um, leftover waste from restaurants, all that food waste. Um, so it actually offers an opportunity for waste management because it then will consume it, generates the larval biomass, right? And then that biomass either can be fed directly to chickens, wet and alive, just like chickens would eat any insect. It can be dried and then fed as aquaculture, as dried individual larvae, and or you can pelletize it. So you dry it out, grind it up, put it in pellets, and then you could feed it to aquaculture as well, or other ways. So what we've been exploring is how do you get that organic matter resource? How do you maintain colonies in Africa using local resources, local knowledge? That local resources and local knowledge will change depending on where you're at, even in one country of Africa, but even in different regions. And so the training program that Dr. Deborah Ummelin has really put together is fantastic, and it does that. It teaches them how to do this farming in their region. And so we want to take that to a continental scale.
And when you say aquaculture, are you referring to like using the insects as like fish food? Like,
Yes.
okay.
Yeah. So a lot of aquaculture will use fish meal. So fish meal that comes from the oceans, usually over harvest the species that are used for that. They grind it, they dry it, they grind it up, and then they throw it into aquaculture to feed other fish. So what we're trying to do is say, what if you replace 50% of that fish meal with black soldier fly meal or black soldier fly product? And does that one affect the health of the fish? Does it make it better or worse? Is the microbial ecology, is it safe? And so those are the types of research questions that we have for aquaculture and for livestock.
Okay. And then last question about Africa. Could you share a bit more or share a bit about your efforts to promote insects as food and feed, and as well as like the recent symposium that you co-organized in Uganda?
So that was the biggest one, and it was pretty recent. We're drafting a report from that symposium on the major takeaways it's intended for African ministers, like Minister of Agriculture, people that can make change and provide resources to push that, and education. So there's extension specialists throughout Africa. And so what we're hoping is if we can draft some standard protocols and teaching materials, that extension specialists can go out and help inform some of the farmers on how to do some of these things. What we learned from that symposium as well is the farmers really understand it sometimes better than the experts, but they need guidance at first, right? And they need some of the scientific, more academic guidance for certain parts of it. But we're learning from them. We're learning what the needs are, where the hiccups are, and so forth. Most recently, I was approached to maybe highlight the successes and the challenges associated with BSF production in Africa, and all what we've been learning from that symposium. And hopefully, I'll be presenting that in India in December. The other way we've been trying to promote this and move it forward is through some partnerships in East Africa and Kenya. That we think we can move with them. We have some opportunities potentially with NGOs because now if you're talking about African small-scale farming, there often are philanthropists that are interested in trying to promote health and well-being, food security, and Black Soldier Fly supports all of that. Unfortunately, Deborah and other partners and I, we had our eyes on some really significant USAID funding, and that's gone.
Yeah.
That was supposed to be my next couple of years in that realm. So we're kind of scrambling to figure out if there's these nonprofit institutes that might be interested because support from the federal government to do work outside of America is challenging right now, unfortunately. But we're optimistic. We think we can do it. European Union has a lot of effort and potential funding sources that might be able to promote this in Africa and maybe India as well.
Okay. I'm sorry to hear that about like USAID. But I wish the best in like securing funding to keep all that going.
Yeah, thank you.
And then last question, I just want to know because I'm from Wayne County. Do you still collaborate with the Wayne County Medical Examiner's Office? And if so, can you tell us what came of it and what some of the key findings were?
That project is pretty much completed. There was some changes in the medical examiner's office there when the University of Michigan gave up on or somehow they didn't sell it, but it was taken over by another university. And so the medical examiner that we worked with was no longer the medical examiner there. We still collaborate with him on some papers that we're trying to get out. But that is such a really cool data set that we have. We have swabs from autopsies, probably 1,500 cases. Some of those that occurred before and during COVID, where what we've been finding is the microbes that are associated with the dead people during the autopsies. There are a couple things that we can determine. One, how long they've been dead, just from the microbial profiles that are collected during autopsy. Two, this is interesting. We get some insight into the antemortem health condition. So before you die, we know that the human microbiome can represent healthy people. It can represent people that have certain types of diseases. And what we think we find in our autopsy swabs is we can actually determine and find bacteria that are associated with someone who's about to have a heart attack or who had a heart attack. And then the most curious one is we can use those microbes statistically, at least, and some machine learning algorithms to predict how they died. So let that sink in. How does a bacterial profile help? How can it be used to predict if you died of an accident, right? Or died of a health condition? I mean, we started getting that out of our data. But what if it could predict pretty well if you were going to be murdered or die a violent death? And our model's pretty good at differentiating some of those. And we couldn't really figure out why for a long time. But then we started working with Amber Pearson in public health. She does a lot of public health stuff. Well, when you live in a high risk area where it's incredibly stressful and your likelihood or the potential risk of being harmed, right? In those areas in and around Detroit, they're known to be at high risk. You have a different lifestyle, a very different lifestyle than, the more affluent guy from outside of Detroit who died of a heart attack because of other health conditions. Your microbiome, and more studies are showing, is a reflection of your overall lifestyle. Some other diseases, deep diseases, but how you live, how your body's treated, the stress that you're under, all affects the bacteria that drive your health and well-being in each human, right? And more studies, I used to say this 10 years ago, but so many studies have shown and supported this in different ways. Not everything, but a huge amount of human health and well-being associated with your microbiome. What we think we're picking up are the signals related to a high stress, nutritionally challenged type of lifestyle. And that is how we're getting these profiles that suggest or are associated with dying by strangulation, by a gun, by stabbing or something like that, which made no sense to me biologically at first. But when you put it into the context of sociocultural economics and sociocultural condition, it's a good hypothesis, put it that way. We don't know for sure, but it's a reasonable hypothesis.
That's really interesting because that's like, I was about to come up with a whole nother tangent, but
Right.
that's really cool. Last thing I want to know is when you were working on the forensic investigations, were you working like directly with like the dead bodies? And if so, how was that? How did that go?
For some of the forensic entomology work, we've gone to the death scene of death.
Okay.
Oftentimes, more often, is the specimens are delivered to you by the law enforcement agency, or the CSI, and then you analyze the data for the insects. A couple of times, we've been able to go to the place where the body's at and do both microbial swabs, but also collect insects, and we published that in the book chapter, I think. But and then when we were working with Wayne County medical examiners, we went down one time to show them how to do the standardized sampling with the swabs. You asked about what that's like. That was eye-opening. There were 11 cases that day, and the efficiency at which the technicians can process for autopsy is astounding, and very, very humbling. At that time, I was 45 at the time, and I walked up and I was like, "What happened to this guy?" And he goes, "Well, he's 45. We don't know. Those are the worst kind. We'll have to figure it out from organs and tox." I'm like, "You don't know." He's like, he just died. I went home and kissed my wife and kids. But I mean, and crime scenes are very different. You to be a CSI, and I get a lot of students that are really interested in forensics and they want to become a CSI agent or technician. Man, you got to have a different mindset. Some of those scenes are gruesome. You have to sit and reflect on your own, your own future at some point, and that's uncomfortable for a lot of people. And then when we go to these forensic conferences where you see people that deal with this every day, from attorneys to investigators to CSI technicians, they compartmentalize pretty well. You have to, or you can't do that job. You let it affect you in major ways. And I'll just say one last thing. I have a split appointment in the College of Osteopathic Medicine. And I don't know, a lot of people know that in entomology. So that's why a lot of this microbe stuff with pathology and the disease ecology work with just microbes sometimes and not insects involved, actually helps me accomplish that part of my appointment as well. I've tried to integrate insects the entire way as well, but sometimes it's just led into microbial ecology of like dead people and how you can use that for public health assessments, even. So that's kind of where we're going with some of that too.
Okay. Well, thank you so much for sharing all that. I really appreciate all the stories, all the information. That's really awesome.